For most of us, blood type is a forgotten line on a medical form. A, B, AB. Oh, numbers and letters we never think about unless we're donating blood or lying on an operating table. But of all four groups, one has a story stranger than the rest. Type O isn't just common. In some of the most remote human communities on Earth, it isn't just the majority. It's practically the only blood type that exists at all. And when scientists began asking why, they uncovered a chain of evidence stretching from frozen Siberian permafrost to the highlands of the Andes. A story of migration, disease, and survival written directly into our DNA. To understand why type O is so remarkable, you have to understand what makes it different in the first place. Blood type is determined by tiny molecular flags called antigens that sit on the surface of red blood cells. People with type A blood carry the A antigen. Type B carries the B antigen. Type AB carries both. Type O carries neither. In a sense, type O red blood cells are blank, stripped of markers that would normally announce, I am a specific kind of cell, treat me differently. This blankness is exactly why type O is called the universal donor. When someone receives blood, their immune system scans incoming cells for antigens it doesn't recognize as belonging to the body. If it finds them, it attacks. But type O cells have nothing unusual to flag. So in most emergencies, they can be transfused into patients of any blood type without triggering the violent immune reaction that would otherwise occur. It's a quiet, elegant trick of biology, and it has made type O blood one of the most valuable resources in every hospital and battlefield on the planet. But outside the emergency room, in the quieter world of population genetics, this same blood type behaves in a way that has puzzled researchers for decades. Because when you map where type O is most common across the globe, you don't find an even scattering. You find astonishing concentrations in very specific places. And nowhere is this more dramatic than in the indigenous populations of the Americas. Across the length of 2 continents, from the Arctic reaches of Alaska down through Central America and into the high plateaus of the Andes, something remarkable shows up again and again in blood surveys. Overwhelming, sometimes near total, dominance of type O. In numerous indigenous communities in the Amazon basin and in parts of Central and South America, researchers have recorded frequencies of type O blood approaching, and in some documented groups reaching 100%. Type A and type B, so common throughout Europe, Asia, and Africa, are barely present, and in some populations, absent altogether. At first, this seems like exactly what population genetics would predict. When a very small number of people found a new population, what geneticists call a founder effect, they carry with them only a fraction of the genetic diversity present in their original, larger population. If by chance, most or all of the founders of a community happen to carry the O allele, and if that community then remained isolated for thousands of years with little intermarriage from outside groups, the O allele could easily become fixed, meaning it eventually crowds out A and B entirely. Add in genetic drift, the random generation-by-generation fluctuation of gene frequencies in small populations, and a strong founder effect can, in principle, produce exactly the kind of near-total uniformity researchers observe. This explanation isn't wrong. It's one of the best established mechanisms in population genetics, and it almost certainly is part of the answer. But when scientists began digging deeper, using ancient DNA to look not just at present-day populations, but at the actual genetic makeup of the people who made the original journey into the Americas, the picture became more complicated and considerably more interesting. The breakthrough came from a source unavailable to earlier generations of researchers: the ability to extract and sequence usable DNA from skeletal remains thousands of years old. This technology transformed population genetics almost overnight, allowing scientists to stop guessing about ancient migrations and start reading the actual genetic record left behind by the people who lived them. When researchers examined ancient DNA from Siberian populations, the ancestral source of the people who eventually crossed into the Americas via the Beringian Land Bridge during the last ice age, they found a genetically diverse population, with type A, B, and O alleles all present, much as you'd expect in any large, mixed human population. This complicates the simple founder effect story. If the population that gave rise to the first Americans was itself diverse, then a total, continent-spanning loss of A and B alleles requires more than one small group happening to carry only O blood. It suggests that whatever the founding population looked like, subsequent generations in the Americas experienced strong pressure that favored survival and reproduction among type O carriers specifically, pressure severe enough and sustained enough to all but erase A and B alleles across an enormous and otherwise geographically diverse landscape. This is where genetics and epidemiology start to overlap, because researchers already understand quite a bit about how blood type interacts with infectious disease, and the interactions are anything but trivial. For more than half a century, researchers have known that ABO blood type isn't just a transfusion compatibility label. It's an active player in how the human body responds to infection. The antigens on red blood cells aren't limited to blood cells alone. Similar antigen structures appear on the surface of cells throughout the gut, lungs, and other tissues, and several dangerous pathogens have evolved to exploit them. The clearest, best documented example is cholera. The bacterium Vibrio cholerae produces a toxin that binds far more efficiently to the surface of intestinal cells in people with type A, B, or AB blood than in people with type O. Multiple epidemiological studies, particularly research following cholera outbreaks in Bangladesh, have found that individuals with non-O blood types are more likely to develop the severe, life-threatening form of the disease. In a strange twist, though, some research has also suggested that once someone with type O blood does become severely ill with cholera, their symptoms can be unusually intense, a reminder that biology rarely offers a clean, one-directional advantage. Malaria tells a similarly complicated but revealing story. Plasmodium falciparum, the parasite responsible for the deadliest form of malaria, causes infected red blood cells to clump together inside blood vessels, a process called rosetting that can block circulation and contribute to severe, sometimes fatal disease. Research conducted in malaria-endemic regions, including studies in West Africa, has repeatedly found that this rosetting is weaker in type O red blood cells than in AB or AB cells, and that severe malaria is somewhat less common among type O carriers. The effect isn't enormous, and it doesn't make anyone immune, but across generations and repeated epidemics, even a modest survival edge can shift the genetic makeup of an entire population. Put these pieces together, and a plausible story emerges, one that doesn't require secret conspiracies or dramatic myth-making, just the ordinary, relentless math of evolution. If ancient populations in the Americas encountered repeated waves of disease, whether pathogens they carried with them from Asia, new pathogens encountered in unfamiliar environments, or a combination of both, and if those diseases interacted with ABO antigens the way cholera and malaria do today, then type O carriers may have had a small but real survival advantage in outbreak after outbreak. Combined with founder effects and genetic drift in small, geographically isolated populations, this kind of selective pressure could, over hundreds of generations, produce exactly the pattern researchers observe. Not universal, but overwhelming dominance of a single blood type across an entire hemisphere. The Andean highlands add another fascinating layer to this picture, though not for the reasons early folklore-style tellings of this story sometimes suggest. Populations who have lived for thousands of years at extreme altitude in places like the Peruvian and Bolivian Andes face a very different survival challenge: chronic low oxygen. Modern genetic research, much of it focused on Andean, Tibetan, and Ethiopian highland populations, has identified real, well-documented adaptations to high-altitude life, including variants in genes like EPS1 and EGLN1 that help regulate how the body manages oxygen and red blood cell production. These adaptations are some of the best-studied examples of recent human evolution anywhere in the world, and they're a genuine testament to how quickly natural selection can reshape a population under sustained environmental pressure. Type O frequency is high in many of these same Andean populations, which is consistent with a broader pattern seen across the Americas: strong founder effects, long periods of relative isolation, and disease pressure layered on top. Researchers studying these populations have also used techniques like ancient protein and antibody analysis from preserved skeletal and dental material to reconstruct the disease history of early Andean societies, revealing evidence of past infections and immune responses that offer a window into what these communities were up against long before European contact. It's a genuinely rich and still developing area of research, though one that requires careful, peer-reviewed interpretation rather than dramatic shortcuts. One of the more debated threads in this broader field concerns Rapa Nui, or Easter Island, and the long-running scientific question of whether Polynesian voyagers made contact with South America before European arrival. This isn't a new or fringe idea. It has been seriously discussed in anthropology for decades, supported by evidence such as the presence of the sweet potato, a crop native to the Americas, in pre-European Polynesian societies. Genetic studies published in recent years, including work analyzing DNA from both living Polynesian populations and the broader Pacific and South American genetic record, have found signals consistent with limited historical contact between Polynesian and indigenous South American populations, likely occurring around several centuries before European colonization. This is a genuinely exciting area of legitimate research, and it's a good reminder of how much can be learned by comparing genetic data across vast distances. But it's also an area where caution is essential. Extraordinary claims about specific shared immune markers or blood type coincidences between Rapa Nui and Andean populations require the same rigorous, peer-reviewed scrutiny as any other genetic claim, and researchers in this field are careful to distinguish between solid, replicated findings and speculative interpretation. It's worth pausing here to say something plainly. The true science of type O blood distribution is genuinely fascinating on its own, without embellishment. Founder effects, genetic drift, and disease-driven natural selection are well-established, thoroughly studied forces, and their interplay across the Americas represents one of the richer case studies in human population genetics. There's no need for hidden engineered defense switches, buried 1998 discoveries, anonymous whistleblowers, or secret deletions scrubbed from public databases. Those kinds of narrative flourishes make for dramatic storytelling, but they aren't how peer-reviewed science actually works, and no credible published research supports claims of that kind. Real discoveries in this field come from painstaking ancient DNA sequencing, careful epidemiological study, and years of replication and peer review. A slower, less cinematic process, but one that has genuinely reshaped our understanding of how indigenous American populations came to look the way they do genetically. What we do know based on solid research is compelling enough. Somewhere in the deep past, small founding populations carried a limited genetic toolkit across the Bering Land Bridge. Over thousands of years, as their descendants spread across 2 continents and encountered new environments, new diseases, and new evolutionary pressures, the genetic dice fell in a particular direction, one that, time and again, favored type O. It's a story about isolation, disease, adaptation, and chance, playing out over hundreds of generations, written in a molecular code we're only now learning to read. Understanding why type O blood became so dominant in certain populations isn't just a historical curiosity. It has real, present-day medical relevance. Blood type continues to influence susceptibility to a range of modern health conditions, from cardiovascular disease to certain infections. And researchers studying indigenous health today sometimes take ABO frequency into account when interpreting broader patterns of disease risk and immune response in these communities. It's also a powerful illustration of a bigger scientific truth. The human genome isn't a static blueprint. It's a living record, constantly being edited by the pressures each population faces, whether that's a pathogen, an altitude, or a journey across a frozen land bridge that no longer exists. The next time someone mentions their blood type as a throwaway fact on a medical form, it's worth remembering that for millions of people around the world, that single letter carries the imprint of a survival story thousands of years in the making, one written not by design, but by the patient, unglamorous, endlessly inventive process of evolution itself. If you found this dive into the hidden history of blood type fascinating, let us know what topic you'd like explored next. There are more stories like this hiding